Serial dilution. Let's understand serial dilution of soil. It's a lab technique we use to make microorganisms, like bacteria or fungi, in a soil sample. Before we jump right into it, I would kindly ask you to subscribe for the channel to stay updated.
Here are the steps for serial dilution of soil. First, take a measured amount of soil and mix it with a set volume of sterile water or a buffer to make a soil suspension. Shake or stir this mixture well so that the microorganisms spread out evenly. Next, take a small amount, like 1 milliliter, from this suspension and add it to another container with fresh diluent. This creates the first dilution, usually a 1:10 ratio. Repeat this process, transferring the same amount into new containers, making each dilution weaker, like 1 to 100 or 1 to 1000. Finally, take a small sample from the diluted solutions, spread it on a culture plate, and let the microorganisms grow. This helps us count or isolate them more easily. Serial dilution of soil is useful for many things. We can estimate how many microorganisms are in the soil, isolate specific microbes for more study, and explore the diversity of microorganisms living there. This technique is super important in microbiology because it helps us work with the many tiny organisms found in soil.